The idea is no longer confined to late-night movies. Consider this: over 20 different species of mammals have already been successfully cloned. This is not a distant future. It is a scientific reality unfolding in laboratories around the globe. We stand at a precipice, looking down at a world where the very definition of an individual could shift. The thought is both fascinating and deeply unsettling. What does it mean when life can be, in essence, photocopied? The questions are multiplying faster than the cells in a petri dish. This journey into replication began with simpler organisms, but science rarely stands still. It pushes boundaries. The leap from a sheep to more complex mammals was a significant one. Each success brought new possibilities and with them, new anxieties. The potential for human cloning, once a taboo subject, now whispers in the corridors of scientific debate. The technology is advancing. The ethical frameworks are struggling to keep pace. Imagine a world where uniqueness is compromised. Think about the implications for society, for individuality, for the very fabric of our existence. The door to a new era has been opened, and we must now decide how or if we should step through it. Cloning, at its core, is the creation of a genetically identical copy of an existing organism. The most common method discussed for mammals is called somatic cell nuclear transfer, or SCNT. It begins with a somatic cell, any cell from the body other than a sperm or egg cell. Scientists carefully extract the nucleus from this somatic cell. The next step involves an unfertilized egg cell. The nucleus of this egg cell is removed. The nucleus taken from the somatic cell is then inserted into this enucleated egg cell. The egg now contains the complete genetic material of the somatic cell donor. Once the new nucleus is successfully transferred, the reconstructed egg needs a nudge to start dividing. This is often achieved using a tiny electrical pulse or a chemical stimulus. If successful, the egg begins to divide and develop, forming an early-stage embryo. This developing embryo can then be implanted into the uterus of a surrogate mother. The ability to clone organisms, particularly humans, throws open a Pandora's box of ethical questions. One of the most immediate concerns revolves around the concept of playing God. Is it right for humans to wield such power over the creation and replication of life itself? Another significant ethical issue is the potential impact on individuality and identity. If a human being is cloned, what does that mean for the clone's sense of self? The potential for exploitation is a grave concern. Could clones be created for specific purposes, such as organ harvesting? Section 4. Echoes of Dolly Milestones and Replication. The world was startled in 1996. News emerged from Scotland of a sheep named Dolly. She was not just any sheep. Dolly was the first mammal to be successfully cloned from an adult somatic cell. This was a monumental breakthrough. It proved that a mature, specialized cell could be reprogrammed to create an entire new organism. Before Dolly, many scientists believed this was impossible. The Roslin Institute team, led by Ian Wilmut, had shattered a long-held biological dogma. Dolly became an instant global icon, a symbol of scientific ingenuity. Following Dolly, the field of cloning research accelerated. Scientists around the world began to apply the SCNT technique to other species. Cattle, goats, pigs, mice, rabbits, cats, dogs, and horses were successfully cloned. Each new species added to the growing body of knowledge. It also refined the techniques, making the process slightly more efficient, though still challenging. Section 5. Shadow Operations. The persistent rumors of secret labs. Alongside legitimate scientific advancements, a darker narrative persists. This narrative speaks of clandestine operations and secret laboratories. It suggests that human cloning is not just a future possibility but a current, hidden reality. These theories often paint a picture of powerful, shadowy organizations. These groups supposedly operate outside the bounds of public scrutiny and ethical oversight. They are said to be pursuing human cloning for nefarious purposes, far from the public eye and regulatory bodies. The lack of concrete proof does little to dampen these persistent whispers. Conspiracy theories frequently focus on the alleged cloning of influential figures. Stories circulate about world leaders, celebrities, or even wealthy individuals creating duplicates of themselves. The motives proposed are varied: to achieve a form of immortality, to provide organ donors, or to create easily controlled political puppets. Section 6. The duplicated dawn. Is our future already copied? The science of cloning has undeniably advanced at a remarkable pace. From the first cloned tadpoles to Dolly the sheep, and now a diverse array of mammals, the technical hurdles are gradually being overcome. Each breakthrough brings us closer to a point where the replication of a human being might move from the realm of speculation to practical possibility. We are already living in an era where genetic information can be read, written, and copied with increasing precision. The tools are becoming more sophisticated, more accessible. This progress is relentless. The ethical debates, however, remain largely unresolved. We grapple with questions of identity, individuality, and the very definition of what it means to be human. The potential for misuse, for exploitation, casts a long shadow over the scientific achievements. So, we are left to ponder a truly thought-provoking question: is the future we once only imagined already here, and has it perhaps already been duplicated?